In this video, I want to show you how to simulate a protein DNA complex using MELD. This video is based on the tutorial on MELD GitHub. We are using 1AZP from RCSB as an example. For simulating a protein DNA complex with MELD, we need a bunch of files, and I will go through each of these files with you. First of all, we need a DNA sequence. This file includes the DNA sequence of only the first strand that appears in the PDB file. So in 1AZP, this is the sequence of the first strand of the double-stranded DNA. Next one is the hydrogen bonds. This file includes all the pairs that are forming a hydrogen bond between the two strands of the DNA. And this is the residue number, the atom name, the residue number and atom name, and the distance that is like the approximate distance of the hydrogen bond. And we are having these data for all the nucleotides of the DNA. We need a file that is called protein contacts. So this is a similar file to the H bonds, instead that this is all the contacts between C alphas of the protein. So this is all internal contacts, and we are using this file to enforce a restraint on the tertiary structure of the protein, because we are not interested in unfolding and denaturing the protein by much. So we are finding carbon alphas that are less than 8 angstroms from each other and are not less than 6 residues away on the sequence. And once we have generated this file, we put it in this format and include it in our directory. We also have a similar file for protein DNA contacts. This includes the residue number of the protein, and we are here using carbon betas. We can use like any atom name. And then we are using the nucleotide number, and then the atom name from the DNA, which is here we are using the phosphorus atoms, then this distance. So once they're less than 8 angstroms, we consider them as being interacting. We also need a secondary structure file for the protein. So this secondary structure prediction is a file that includes a character for every residue of the protein and also the DNA. So the first few residues of the DNA are represented as periods here. After that, for the protein, wherever we have a coil, we are representing that residue with a period. Beta strands are represented by E, and helices are represented by H. This file is read by MELD, and it includes the secondary structure prediction for our protein. And remember, for every nucleotide of the DNA, we are including a period at the beginning of this sequence. And then we have a restraints.py. This file includes some functions and classes and everything that are required for defining restraints in MELD. We usually don't need to modify this file, so we just include it in the directory, and this file is used by setupmeld.py to generate the restraints based on the other files that I just explained to you. And last of all, it's the setupmeld.py. This file includes all the parameters that are required for setting up a simulation in MELD. So let's go over this file very quickly and then run our simulation. So first thing first, we will need to import a bunch of modules. Most of them are from MELD, some of them are for OpenMM and NumPy and etc. We are also importing that restraints that the other file I just mentioned. We need to define the number of replicas. So remember, in MELD we are doing temperature and Hamiltonian replica exchange. So here I'm using 30 replicas, and I'm using 20,000 steps and a block size of 100. So it means like every 100 steps it will write the output into file. Then I will use this function to generate a template. So here I'm using the FF14 SD side, and then I'm building a molecule from PDB file. That PDB file is in my directory. I'm making a system from that molecule that I built, and I'm also defining its coordinates, and I'm also defining velocities, initial velocities, and also alpha. So alpha is a parameter that scales from 0 to 1. 0 corresponds to the lowest replica, and 1 corresponds to the highest replica. And then we will use this setup system function to generate our system. First, we have a templates. So templates will look into the template directory inside our directory, and it will use whatever PDB files that are in there. So in this current example, I'm using only one PDB file, but you can use whatever number of PDB files that are less than your number of replicas, and those are used as seeds for your simulation. So you can have like multiple PDB files which have the same number of atoms, and they will fill up the replica exchange. Here I'm defining a temperature scalar. So this temperature scalar will scale the temperature from 300 Kelvin to 500 Kelvin, from replica 0 to replica, from alpha 0 to alpha 0.5, which corresponds to replica 0 to replica 50. And then I'm defining a protein scalar. This protein scalar is using a constant, force constant, and I will apply this protein scalar on protein contacts like that. So I'm using the contacts that I defined in protein contacts, and I'm using this scalar, and here I'm defining a string based on the scalar and this contact file. And I'm multiplying it here by 0.9 because I just want to trust 90% of this contact file, and allowing 10% of those contact files not to be satisfied, so that I'm actually allowing some flexibility in the tertiary structure. Next, I'm giving it the DNA sequence, and from the DNA sequence, it will generate the H-bonds file. So remember, the H-bonds file is generated as part of the setup node.py. I don't need to generate it beforehand. And then I'm using the same scalar, protein scalar, to define restraints for the hydrogen bonds. I'm also using the same protein scalar to restrain the Cartesian coordinates of the DNA. In this example, we are not interested in conforming the DNA or separating the two strands from each other or bending it. So we are keeping the DNA in a similar conformation to its starting structure. Next, we define a secondary structure scalar. This is again a constant scalar. We could use like the protein scalar again. And here we are giving it the secondary structure at that file. And we are also giving it the force constants for torsions and distances. And we are defining this, this restraint here. Here we are only trusting 96% of the secondary structure prediction. And last but not least, we need to define restraints for protein DNA contacts. So in this example, we are defining two sets of restraints. In the first set, we are using the protein DNA contacts file. So this protein DNA contacts file, as I showed you, includes all the pairs from protein and DNA that are interacting. This scalar is not a linear one, it's not a constant scalar, so it's only present from alpha 0.4 to alpha 1, and it scales from 0 to 4. So at low replica, the force of this scalar is minimized, and high replica, it's stronger. And so we are applying this again, and you can see that below 0.4, this scalar is not in effect. So we are only sampling by forceful forces. And here we are defining a second set of restraints. Here we are selecting all the carbon betas in the protein, and also all the phosphorus atoms from the DNA, and we are restraining the center of mass of those two groups to each other. This set of restraint is only active from alpha 0.7 to alpha 1, and here at alpha 0.7 we are pushing those two central masses to 0.5 nanometers from each other, and at alpha 1 we are pushing them at 7 nanometers from each other. So the idea is that we want to make sure the protein and DNA separate from each other at high replica. And so here we are defining the restraints again. We are giving it a scalar and the force constant. The two groups that we are defining the COM restraints between them, and the distance is defined by this positioner, and the weights are set to none. And then we append this restraint here. At the end of this file, we have some options. These options are usually not needed to be modified. They are some parameters for the simulation. So here we are using hydrogen mass superposition, so we can go to to a long time step of 4 femtoseconds. We are using the GBNet2 implicit solvent model. The cutoff is 1.8 nanometers, and the number of time steps, and we also have minimization before the replica exchange actually begins. And here we are also storing all of those parameters and all of those restraints, and then later when this function is called, it will generate some files that are used by OpenML to run the simulation. I will also show you in my directory here. So in this directory, I have starting a structure. I will show it to you. I've separated these, the protein and DNA, from each other. And you can see there is a distance between the two. And once the simulation is done, we expect that we observe multiple binding and unbinding events to happen between the protein. All right, in my directory now, I have all the files that are necessary for running the simulation. I've already loaded MELD, and by running the setup script, these warnings are normal. By running this script, it will generate some files that are required for OpenMM to apply the restraints that we defined. So once this script is run, we have a new data directory. In this data directory, all the files that I just mentioned are generated. Once the data directory is there, we can go ahead and submit a job for running the MELD simulation. So here I'm using the University of Florida's Hypergator. I'm using a standard setup script to submit my job. And once I submit it, it will go into the queue for running. To show you some of the outputs, I will use another directory that I have already run. So this is a trace of the replica ladder. So we have from 0 to 30, and each color represents a replica walker, and you can see that they have a good exchange along the simulation, and we expect the replica exchange to have frequent exchanges between the replica. I can also show you the last steps of each replica. So I'm putting them all on top of each other. And you can see that I have like every replica here. And this is actually the last step of each replica. And you can see like these ones that are unbound here correspond to the high replica, and the ones that are bound to the DNA at the center, they represent the low replica. For example, if I remove this 29, 28, 27, these are all unbound because we applied those restraints and those forces that would separate the protein and DNA